stem cells and fermentagenesis. Frieda.
Thank you. So today I'll be presenting my research in the metabolic influence of spermatogonial stem cells. So for those of you who were at CRB and or heard Dr. Oatley's talk earlier this year, some of the material you might have heard of, but there are exciting new stuff in the end of it. So why do we care at all about spermatogonial stem cells? Infertility affects approximately 10% of reproductive age couples worldwide, and half of those cases are attributed to the male factor. So if we want to put it in a much more relevant manner, this room fits about 100 people. 10 of us will suffer from infertility, and 5 of us are boys. So most of most of the cases are idiopathic with no known causes, but some of the known causes are attributed to side effects from metabolic syndromes and cancer treatments. Gonadal failure and sterility are two of the severe side effects from cancer treatment. While adult males can cryopreserve their sperm to preserve fertility for later, prepubescent boys currently we do not have feasible ways to cryopreserve the sperm and or induce fertility afterward. So within the past few decades, the juvenile cancer patient survival rate has increased to about 80%, which means that 1 in 250 people about our age are now suffering from infertility and are long-term cancer survivor. So within the Pullman campus, we have approximately 25,000 students. That means 100 of us are long-term cancer survivor who are suffering from infertility. So therefore, it is very important for us to understand spermatogenesis so that we can preserve the spermatogonial population during cancer treatments either via in vivo or an in vitro method. This is a diagram of mammalian spermatogenesis. To be more specific for the mouse. So spermatogenesis involves the self-renewal of the spermatogonial stem cell pool, which then proliferate. It amplifies, and then they commit to differentiation. And then through various steps of meiosis, it will go through into sperm. And if there's interruption or imbalance in any of these steps, you will have infertility. During cancer treatments, the chemotherapy and radiotherapy agents are targeting for this population, the undifferentiated spermatogonia population. So it is very important to study the ground states of spermatogenesis so we can develop methods to protect the spermatogonia. So this picture shows a spermatogonia population that includes the darkened differentiating spermatogonia and the undifferentiated spermatogonial population. Today I'll be focusing on the undifferentiated spermatogonial population. This population includes the stem cells, which is a subpopulation of the asingle cells, and then it amplifies in number before committing to differentiation. And this population of spermatogonial stem cells maintain the stem cell pool, and if there's interruption to it, again, it will have infertility. But unfortunately, we do not have a marker currently for the stem cell population, so we cannot understand the molecular characteristics of the spermatogonial stem cells. Although we don't understand the molecular characteristics, we do know the functional characteristics of spermatogonial stem cells. So let me start by defining what stem cells are. Stem cells, they have to have regenerative capacity, and they have to sustain a developmental cell lineage. So for spermatogonial stem cells, they have to regenerate spermatogenesis and sustain spermatogenesis. Whereas the transiently amplified progenitors do not have regenerative capacity and only contributes to the stem cell states. So it is very important for us to distinguish a molecular marker for the spermatogonial population so that we can understand its molecular characteristics. To do this, previous recently my lab has identified a protein called inhibitor of DNA binding for ID4 as a marker for spermatogonial stem cells. If you look at the cross-section of the germ cells, here in panel G, you can see the red stain is ID4, and then PLCF in the middle, panel H, stains for the undifferentiated spermatogonial population. And panel I, here is a blown-up picture of the merge image. You can see that ID4 stains by subpopulation of the PLCF undifferentiated spermatogonial population, indicating that ID4 is a putative marker of spermatogonial stem cells. So, to further investigate ID4 as a marker for the spermatogonial stem cell, an ID4 GFP reporter mouse was generated. The GFP coding sequence was inserted in frame into exon 1 of the ID4 gene so that GFP expression is directly proportionate to ID4 expression. The ID4 GFP protein expression is found in testis, brain, and kidney, which is also where ID4 proteins are found. These transgenic reporter mice are then crossed with a Rosa mouse. Rosa mouse has the LAC-Z transgene, so that in the presence of X-gal, the LAC-Z expressing cells will turn blue. So by crossing these two mice, we can generate a culture of heterogeneous undifferentiated spermatogonial population that has stem cells and progenitor cells. These cells not only has ID4 GFP that is about 10% of the population, which is proportionate to how much stem cells is usually in previous undifferentiated spermatogonial culture. This culture can also be used in a functional transplantation to recipient testes that do not have the LAC-Z gene, so that all transplanted testes, transplanted cells, when they regenerate spermatogenesis, will turn blue. So with this tool, we then wanted to separate out the ID4 GFP positive population to investigate its regenerative capacity. As you remember, stem cells has to have regenerative capacity. Using fluorescence activated cell sorting, we separated the ID4 GFP population from the ID4 GFP negative population. These cells are then transplanted into a recipient's testis. The recipient does not express the LAC-Z gene. They also are devoid of germ cells. So if there is spermatogenesis, they are truly from our donor cells. With 5 different biological replicates and 20 different animals, so 40 testes, 20 for the GFP positive and 20 for the GFP negative, we see that the ID4 GFP population on the left is definitely enriched in spermatogonial stem cells. Each of these blue colonies, as you can see there, is spermatogenesis generated by one spermatogonial stem cells, whereas the ID4 GFP negative population has nothing, which indicates that these are the progenitor cells within the culture. So knowing that ID4 GFP positive cells are now the marker and are the spermatogonial stem cells, we wanted to further investigate the transcriptome of this population. So to do this, we again used fluorescence activated cell sorting to separate the two cultures. With 3 different biological replicates, we utilized the Illumina platform of RNA sequencing. After analysis, I found that there was no difference in expression of approximately 10,000 genes. And then looking at my data, the ID4 GFP negative progenitor population has approximately 25 genes that are upregulated while they are downregulated in the stem cell population. And then if you look at the ID4 GFP positive stem cell population, there are 12 genes that are upregulated in the stem cell population but downregulated in the progenitor population. These 12 genes include genes for piRNA metabolism, cell growth and metabolism, intracellular signaling, and transcription. So looking at these 12 genes, 2 of them really hit home to me and are very important to me. As you remember in the beginning of my talk, I mentioned that some of the known causes of infertility are associated with the side effects of metabolic syndrome and also cancer treatment. So enolase 1 and 2 transcripts generate proteins that are involved in glycolysis. Glycolysis is a metabolic pathway. Additionally, a lot of cancer cells utilize glycolysis, so a lot of cancer drugs are focusing on targeting enolase 1 and 2 or other enolase proteins. So for me, understanding the functional and biological relevance of enolase 1 and 2 within the spermatogonial stem cell population is very important. So from now until the end, you get to hear about this. So to familiarize you with what enolase 1 and 2 does, enolase 1 and 2 performs the penultimate reaction in glycolysis. It converts 2 phosphoglycerates into phosphoenolpyruvate, PEP. It generates the second of 2 net gain in ATP in glycolysis. And most importantly, glycolysis is the preferred pathway, metabolic pathway of most tissue-specific stem cells. So I wanted to validate my RNA-seq data. To do that, I utilized RNA extracted from the positive and negative population, generated cDNA, and then using real-time quantitative PCR, you can see that the positive population definitely has an increase in upregulation of the transcript, 17-fold for enolase 1 and 11-folds for enolase 2. Seeing that the transcript is upregulated, I wanted to investigate if the proteins are also translated more often. To do that, I used Western blots. As you can see, the positive population is here, negative is right here. You can see the graph on the right-hand side shows the quantification. Enolase 1 is about a third less in expression, while enolase 2 is much more apparent, even from just the blood, that there's a lot more enolase 2 concentrated in the stem cell population, whereas the progenitor population doesn't really express enolase 2. So this is very important and very interesting to me. So I wanted to utilize my culture of the ID4 GFP laxi mouse. Again, this is a heterogeneous culture of both stem cells and the progenitor population. I wanted to treat the culture with sRNA, which will transiently reduce the transcript abundance of enolase 1 and 2, and then also control. With this treatment, I will be able to decrease the amount, not only transcript and hopefully the protein abundance of enolase 1 and 2, then I'll investigate what will happen to my stem cell population. I utilize gene expression analysis, protein expression analysis, ID4GFP contents analysis with flow cytometry, and a functional transplantation analysis. So first, after approximately 24 hours of transient reduction, I collected RNA using real-time quantitative PCR. I found out that the transcript abundance in my treatment culture for enolase 1 has a 70% decrease, while my enolase 2 transcript abundance in my treatment culture is approximately 40% decrease. Seeing the transcripts decrease, I wanted to know if the proteins abundance also decreased. In the left is 24 hours treatment of control, and then on the right is the 24 hours enolase 1 and 2 transient reduction. And here is the quantification of the Western blot, and you can see there is slight decrease in enolase 1 and 2 protein. Again, these are preliminary data. They will be optimized. So with slight decrease in enolase 1 and 2 protein, I wanted to see if that will affect my stem cell abundance within my heterogeneous culture. To do this, the quickest way, as you know, the culture, all the stem cells express the ID4 GFP. So using fluorescent flow cytometry, I can measure if there's a decreased amount of ID4 GFP cells. After 24 hours, there's no significant decrease in the GFP content, but at 48 hours, there's about 25% decrease in the ID4 GFP cells abundance. So with this in mind, I wanted to ask if the decrease in the fluorescence is proportionate to the decrease in stem cell capacity. So to further investigate, this is currently being performed. I treated the cells for 24 hours and then let them grow for about a week. And then after that, I am going to transplant them into recipient testes that are devoid of germ cells. So just to remind you, the stem cells, each of them, if they colonize, it will turn blue. So I will measure how many colonization and if there's a decrease. And that will be later for you to find out what will happen. So for future directions, why would I want to find out enolase and metabolic pathways influence on the Spermatogonial population? Well, this is a picture of very basic, rudimentary bioenergetics of a mammalian cell. Within the mammalian cell, there's glycolysis, beta oxidation, citric cycle, and oxidative phosphorylation. And my hypothesis, if you're interested, from my preliminary data, that will be in my second proposal, is that the Spermatogonial stem cell population and progenitor population have difference in bioenergetics, means that they will utilize different metabolic pathway for the preferred energy source. So Spermatogonial stem cells, that population, the ID4-GFP population, would prefer glycolysis, while the progenitor population, the ID4-GFP negative population, would prefer beta oxidation. If you're interested in it, please come to my second proposal. So how will this contribute to my field and also other fields? So with understanding how to culture and what is preferred by the Spermatogonial population, we can have novel strategies in protecting and preserving the Spermatogonial population during cancer treatments, either in vivo or in vitro. There are no ID4-GFP humans walking around, but if we can develop ways to enrich for Spermatogonial stem cells in culture, we can then translate that to human culture and transplantation, because currently we do not have a way to culture for human cells. And at the same time, after we take a biopsy, we can, during cryopreservation of germ cells, we can favor for the Spermatogonial stem cell preservation and maintenance during cryopreservation. So finally, the information from this project can help contribute to other tissue-specific stem cell lineage. So for example, in hematopoietic stem cells, they currently have no way to culture and proliferate for a long period of time. So this will also help. So thank you very much. And I would like to thank my lab, my committee, and everyone at SMB, the graduate students, faculty, and staff, and additionally, very important, the funding from NIH. Thank you very much.
Questions for Frieda?
Yes.
Is there evidence in any other system that glycolysis is regulated by transcriptional control of enolase?
Not that I can see in the other tissue-specific stem cell systems. So, not within any of the, yeah, I have looked at stem cells, I've looked at hematopoietic stem cells. So
Or in any other model system, any other model organism, or anything like that?
I'm not familiar with it, but that's an interesting question. Yes?
So there may be an alternative hypothesis that you should consider. You should give consideration to the new knowledge that a lot of these endomyroharness pathway enzymes have alternative functions. So if you are seeing a particular phenotype, that phenotype may not be because something's happening to the enzyme, but because something's happening to the alternative function. For example, in humans, enolase is also found on the surface of liver cells. Enolase is secreted, it becomes surface associated, and other things happen that are non-enzymatic in nature. In both prokaryotic and eukaryotic organisms, about half of the Embden-Meyerhoff-Parmas enzymes also are secreted. They become surface-associated and now have completely non-enzymatic alternative functions. So I think that's something that you should at least think about.
I would definitely consider that. Yeah.
I'm curious, what do you think it is about the stem cell niche that would provide an environment that would preferentially prefer glycolysis as opposed to what regenerative cells are doing?
So his question is, within the basement membrane of the stem cell niche, where both the stem cells and progenitor resides, why would one population prefer glycolysis whereas the other population prefers beta oxidation? Is that correct?
Yeah.
So I actually have investigated a little bit into that, and so I do not believe that there's a hypoxic or differential oxygen environment between the two populations. And you will then question why would prefer glycolysis over beta oxidation if there's not an ambient change in oxygen level, if they're all inside the basement membrane. So to preserve the fidelity of DNA, and the cells are usually slow growing, most tissue-specific stem cells, in opposite to embryonic stem cells, which can divide much faster. And so with that in mind, if you have much more ATP production through the oxidative phosphorylation pathway, you have a lot more reactive oxygen species, which will then interfere with DNA replication or the fidelity of the DNA population. And because the stem cell population is so important that it actually contributes not only to all the sperm that will come out, it also will contribute genetic information to the next generation. So to have less amount of ATP produced is probably preferred. So that's why I think glycolysis is the preferred pathway. But then when you change into progenitor, you're transiently amplifying, as you saw in the chains and the aligned cells, that goes up to 16. And to have so much growth and mitosis going on, you need a lot of ATP production for cell growth. And in my progenitor population, 25 genes, there are a lot that I didn't talk about. There's about 5 genes that protects the cell from the reactive oxygen species that are associated with the mitochondria. And then there are also DNA packaging genes too. So that's why I came to my hypothesis.
I have a
Yes?
technical question. How long do you wait after you've back sorted the cells? Do you just take them straight and do your analysis to look at the different transcriptional regulations, or do you grow them in culture for it?
So that's a very good question. So after we back sort the cells, do we use them immediately or do we culture them? So excellent question, because I didn't include a lot of things that are associated with that. We use them immediately. Because if you culture them, what I have found, and what our lab has found, is that if you culture them, some of the stem cells will start to want to proliferate and change into progenitors. And then, so that's very important that we use the population immediately. So we do fact sorting, and then immediately analyze the transcriptome. Yes?
Along those lines, in your knockdown, how stable is the knockdown, and would it carry over to the transplantation process?
So your question is, when I use the transient knockdown of enolase 1 and 2, how effective and how long will it last? So for siRNA, the transient knockdown is approximately, it finishes at 1 week. So what happened is, I am trying to reduce the amount of enolase, so inhibiting the growth. The growth of our cell line is about a week it takes to divide. And so if I inhibit the enolase population, I'm inhibiting the growth of that population. So that when I transplant it, so if I have 10 cells to start with, if, say, they didn't grow, the control population will grow and become 20 and double in size, the 10 might remain at 10 or go down because they're unhappy because the preferred pathway is gone. Then when I transplant it, I might be transplanting 8 as opposed to 20. So then when I analyze it, then the recipient testis or the control will have 20 colonies, whereas the treatment will have like 8. So that's how I'm going to analyze my data. Does that answer your question?
Other questions for Frieda? If not, let's thank her again.